today is about. And so we're going to start by talking about what we call the central dogma of cell biology. Central dogma. Does anyone know what the word dogma means? Dogma is something that you sort of believe as the center of your understanding of things. So we talk often, you'll hear it used in reference to religious people being dogmatic, where the idea is that sort of their religious beliefs form the center of their thinking, which for the record, you believe in a religion, I think that makes sense. In this case, the central dogma of cell biology is this. It kind of comes in two parts. And we're going to write it in sort of plain everyday language, at least I hope. Part one of it is just that all living things are made of cells. And then the second big idea is that every cell is the offspring of a previous generation of cells. You could apply a similar line of thinking to human populations, a similar sort of central dogma. Every person is a human, and every human is the offspring of a previous generation of humans. Here we're talking about cells themselves. So the idea being that out in the world around us today, none of the cells that we come across randomly came into being. They didn't form by lightning striking, a pond or something like that. Every single cell on Earth exists because a previous generation of cells reproduced. That kind of makes sense? Which means that cells must have a mechanism by which they reproduce, right? If things are going to reproduce, they have to have a mechanism by which they do so. If you take grade 11 biology, we'll learn a little bit about how reproduction happens on like the multicellular organism scale. We'll talk about a reproductive system a little bit. You probably learned about that in grade 9 health, I'd imagine, right? How your reproductive system works, or grade 8 health. I have no idea when they teach these things. But hopefully at some point they've sat down and explained to you how your bodies basically work. But we're going to start with a much narrower view of things, which is not how a whole big animal reproduces, but how just a single cell reproduces. That's going to be the focus of our lesson today. So every cell has a life cycle. And single-celled organisms single-celled organisms must reproduce to survive, whereas in multicellular organisms like you, what does multicellular mean? What does multi mean as a prefix? Multiple, yeah, which means lots, right? More than one. Multicellular means lots of cells. You're made of lots of cells, not just one. In multicellular organisms, some cells reproduce and some don't. Your neurons don't reproduce. In fact, in humans, interestingly, most of your cells that actually do stuff don't directly reproduce. We have a whole set of cells whose job is just to reproduce. So your skin cells don't make new skin cells. Your skin cells just die. And then cells underneath there produce new skin cells, which is kind of interesting. Your nerve cells, as a rule, don't reproduce or get replaced at all. Just if they get damaged, they tend to stay damaged. Which is a disadvantage, right? Like if your nerves could heal the same way that your skin heals, that would be an advantage. But that's something our things don't do. So what does this look like? Let's look at a typical cell life cycle. And this is going to be low detail. All right, the low detail version. I sometimes like to sort of teach things 2 or 3 times. And the first time I teach it, we explain it like super simply. And then the second time through, we explain it in a little bit more detail. And then maybe in the third time through, we explain it in all the detail. So the basic version of it is like this. We have a new cell. And we're going to introduce some structures here that you may or may not be familiar with. So what we have here is this whole thing is a cell. Some teachers, and this is not a bad thing, there's nothing wrong with this, just a different approach. Some teachers like to give you a handout, and here's the cell, and they put a diagram of it, and they label all the parts, and you list what they do. I've never done that, because in my mind, it doesn't really breed understanding what they do. You just sort of write it down and then move on. So I always like to teach the parts of the cell by teaching what they do, like in an actual explanation of what they do, not just by putting it in a sentence and then moving on and being like, the Golgi apparatus is like the cell's post office. And you're like, what the hell does that mean? I'm like, don't worry about it. Let's move on to the next thing. Just my preference. This structure in the middle of the cell, this is a word that we use to mean the structure in the middle of pretty much anything. So you're probably familiar with it. We refer to this as the nucleus of the cell. Does anyone know what's inside the nucleus of a cell? And by the way, when you look at cells in a microscope, if they're bigger cells, even with just a basic light microscope, you can usually see the nuclei. You can see the nucleus of a cell. Does anyone know why we have a nucleus inside of a cell? It contains something. It contains probably the most important thing in the cell. It contains what we call your genetic information. Does anyone know what that means? Does anyone know? I mean, we're going to write it down in a sec, but you've probably heard of this before. DNA? You heard of DNA before? Okay, what's your DNA? Every cell has it, or RNA. We'll talk about the difference between them later. And you've probably heard this word before, genetics. What are your genetics? Maybe you've heard genes. Does anyone know what that means? Well, that's okay, because we're going to write it down in a second. And then this actual material that surrounds the cell, the outside of it, we call that the cellular membrane. And again, don't worry if you haven't heard of any of these things before, because we're going to talk about it right now. We're going to say a cell is a huge collection of molecules that serve all sorts of different functions. When we draw a cell, we don't draw these because these molecules are so small you can't see them even with the most powerful microscopes on it. Individual molecules are very, very, very small. But they float around in a fluid called cytoplasm. And we're going to start learning how to kind of break words down in science, because this prefix cyto always just means cell. And this term plasm or plasma refers to liquid. These both come from Latin. So this word may sound fancy, but if you spoke Latin, they float around in a fluid called cell liquid, is what that sentence would say. Right? It's not fancy. Scientific words sound fancy. They're actually just really dumb. They're exactly what a grade 2 kid would come up for naming things. They're looking at a microscope. They're like, the cell, it's full of a bunch of liquid. What should we call it? I've got a great idea. Cell liquid. And then they wrote it down. The reason we use Latin is because people study science in a lot of different languages. And so scientists have tried to agree that even though scientists all speak different languages, the word we use for things in science, we try to have be language neutral. So a French scientist still calls this cytoplasm, and a German scientist still calls this cytoplasm. We've all agreed to use this old dead language Latin that nobody speaks anymore so that we're using the same words for things. Does that make sense? Now, to be clear, was this still obviously very much decided by a whole bunch of white guys who'd been taught Latin at their schools? Yes, it was. So don't worry. If you're worried that this sounds really egalitarian and fair, still very much a product of sort of the English education system. But how does that feel? Not too bad? By the way, we didn't draw it in this diagram, but that's this fluid here. Just the fluid inside is called the cytoplasm. A membrane, which is just a tough but flexible I almost want to use the word tissue, material, completely surrounds the cytoplasm. Again, membrane isn't a word specific to cell. Membrane refers to any thin, flexible material that separates two things. So for example, when you're building a speaker cabinet, if you want to isolate bass, you put a rubber membrane between the speaker and the back panel. What else? If you're anyone ever done any roofing? If you're doing roofing and you want to do a good, more expensive job, you can pay extra money to have a rubber membrane put down over the plywood on your roof before you nail down the shingles so that there's an extra layer of protection against water getting into your roof. So membrane just refers to any flexible material that's used to separate two things. But in the center of the cell, it's not in the center. That's true. That's not true. It's often in the center, but not always. In the cell, another membrane surrounds the most sensitive material. In the cell. The DNA, or in some cells, RNA, which is the genetic material. And again, we'll talk about what that means in a second. You can just write it. So again, if we're redrawing our cell, there's our cell. And here we have what we call the nuclear membrane. And again, that just means the membrane that surrounds the nucleus. The whole thing is called the nucleus. The fluid inside that is called, not cytoplasm, cell liquid, but how would we say the liquid inside the nucleus? Nucleoplasm. So that is the nucleus liquid. And then inside there is this molecule called DNA, which, when you look at it under a microscope, just looks like a hazy, stringy kind of bit. If you take grade 11 biology, one of the things we do is we actually extract the DNA from usually strawberries. I've done it with bananas as well a couple of times in the past. But it just looks very stringy and sort of filamenty. Well, that's not what I wanted now, is it? We'll learn specifically how the DNA or the RNA does this if you take grade 11 biology, and then in even more detail if you take grade 12 biology, both of which I think are great classes. But all you need to know for now is that this long chain of a molecule is basically coded instructions. It's a blueprint. And specifically, it's the blueprint to build and operate this cell. Does that make sense? It actually has a coded piece of information in it, and literally it has its own alphabet. So just like we write information down by putting down alphabetic letters, and those can be read as words, and you can read those as instructions, that's how all these sentences are working, right? Your DNA is a big, long document, essentially, that has its own alphabet, and it is a set of instructions on how to build a cell and how to operate that cell. And if you're a multicellular organism, it's actually the instructions on how to build all the cells and how to operate all the cells. It's an enormous volume of information. It's crazy. You have your genetic code is like megabytes of information stored inside each one of your cells. It's pretty crazy. So the DNA / RNA is a molecule that contains coded instructions for building and operating the living organism's cells. Possibly cell if it's singular celled, and possibly cells if it's a multicellular organism. And as you can imagine, why does this get its own second little membrane inside the cell? Its own membrane, its own nucleoplasm, kept separate from everything else? Why do you think? Well, if you damage the instructions on how to build and operate your cell, what happens? Well, then the cell wouldn't know what to do, yeah. Yeah, it could potentially break. In fact, we have a word for it when that DNA gets damaged. If your DNA gets damaged and some of the instructions get changed, we call that a mutation. Very good. That's right. We actually call that a mutation. That's a very serious deal. So the DNA must be protected to avoid damage. It also has to be read. So those machines whose job is to read the DNA and then make a little copy of it that gets shipped out into the rest of the cell. And so in the main cell, they're always working with copies of the DNA, but never with the original, which is a pretty smart way of doing it if you think about it. But it's also kind of similar to how do we do things, right? Like we print off copies, this or that, but we tend not to, if you wanted to study the U.S. Constitution, you're going to be looking at a photograph of it or a scan of it. They're not going to be like, oh, Orlando's class is studying the U.S. Constitution again. Let's go get the U.S. Constitution that was signed by all the founding fathers, right? They're not going to do that, right? And if you wanted to study, anything else, they don't give you the original thing to work with, right? That wouldn't be very safe. They give you a copy. Most museums, the really, really, really delicate artifacts are either kept away in a glass display case like that, right? Their own little nucleus, or often it's a casting of them. It's a copy that's been made, and the original is kept safely in the back. I know one of my dreams in life is to go to Cairo and go to the Egyptian Museum in Cairo, which is, house to many, though interestingly not all, because the British, boy, they stole a lot of those artifacts, but home to many and still won't give them back. But you can visit a lot of these things, but a lot of the things on display there are actually reproductions, and the original is kept back where people study them and they're kept safe. Though that's a relatively new development. One of the interesting things is, for example, if you go to Egypt, still to this day, you can actually go in all of the real tombs. You can go in King Tutankhamun's tomb, and you can go in the tomb of Ramesses II and Hatshepsut and all these things. And of course, with people doing that over the years, they become quite damaged. So there's talk about building a replica Valley of the Kings, where they sort of make copies of all these things that tourists could go into without damaging the originals. Anyway, in the cell, the original DNA, the original information is always kept safe in the nucleus. And if they have to work with it, they work with copies. Okay, why do we care about this? Well, now we'll get back to that life cycle. In stage 1, the cell is just doing its own thing. We call this interphase, which just inter means between, right? And so the reason we call this interphase is because it's kind of between noticing anything interesting going on vis-a-vis reproduction. So in interphase, the cell is just doing its thing. This is not a very scientific explanation, but I'm going to put it anyway. Cell's just doing its thing. If it's a muscle cell, during interphase, it's just being a muscle cell. It shortens, it lengthens, it shortens, it lengthens, and it's just that. If it's a amoeba, it's floating around doing its amoeba thing. It's eating its dinner and whatever. If it's a bacterial cell, it's swimming around with its little tail and eating food and whatever, making people sick. So in interphase, the cell is just doing its thing. This is, in other words, normal cell life. Some cells will spend years in interphase. Your neurons spend their entire existence in interphase. They never reproduce. So, and you might live to be, 150 years old. I don't know. Nobody's done that yet, but you could be the one. Certainly, if you're the average Canadian woman, you will live to, is it 84? And the average Canadian man, like 81, I think, something like that. Those cells will spend 80 years in interphase, 90 years in interphase. So this can be for very different lengths of time, depending on the type of cell. Does anyone know how quickly bacteria tend to reproduce? Some bacteria can reproduce as often as every 20 or 30 minutes, which is why, if they're under ideal conditions, which is nice warm temperatures with lots of moisture, which is why we don't leave our food out in nice warm conditions with lots of moisture, because we don't want those bacteria on everything, unavoidable. You keep your food in the fridge, those bacteria are still reproducing. You know that eventually your food will spoil, right? But they do so fairly slowly. You leave it out sitting on the counter, you're just giving them, you're saying go nuts. So there we go. And then, near the end of interphase, the cell makes a copy of its DNA. So you can imagine it looking like this. We have in here, and we had all the DNA, right? There it is. And what's going to happen is the cell is going to have some machines in its nucleus whose job is to take that DNA and make a copy of it. So if that's the DNA, this green one here is ideally a perfect copy of the DNA. I say ideally because sometimes mistakes happen. And we already said what those are called, right? What do we call it when something gets changed in the DNA? A mutation, that's right. But we're not going to deal with mutations yet today. I'm not happy with where I put that. If you're not happy with where you put it, you could just highlight it with your pen like that and just move it. Doesn't that look better? That's better, right? Okay. And then, why does it do that near the end of its life? What is it getting ready to do? Sorry? Yeah, though cells don't, well, cells do sometimes die. But what does the cell want to do before it dies? Want to. I'm ascribing as if it has a brain that's thinking, which it doesn't. What do cells do before they die? Generally, not every cell, but certainly some, since there's still cells around. Right? If the cell just lived its life and then died, you wouldn't be here. So cells have to do something before they die. Yeah. Very good. They have to reproduce. And that's stage 2 of the cell life cycle, reproduction. And we call this process mitosis. Have you heard that word before? I don't think I had before. Actually, no, I know that I had before I went to high school because my sister is older than I, and I remember her complaining about having to learn it, actually. I have a vivid memory of that, yeah.
I heard it in Twilight.
Oh, there you go. Is it like a classroom scene? And they're learning about mitosis? There you go. I can live with that. Is that the first one?
Yeah.
I shouldn't tell this story. I went and saw that film with my friend Jay, but I missed a memo. I'm not really a drinker. I don't drink as a rule. And so my friend Jay had sent an email saying, we're all going to go see Twilight. And I thought, that's odd, but okay, why not? And my friend Jay worked at a movie theater, so he liked to have fun. So I went to see this movie Twilight. What I missed was the memo that they had all planned to drink a real lot beforehand and then be really obnoxious before the movie, which they proceeded to do. And then they all got kicked out for like heckling at the movie, which resulted in me sitting alone at a movie theater by myself watching Twilight as like, I don't know how old I was at the time. That movie's what, maybe 12 years ago? Does that sound about right? I think I was roughly 30 is the point. What I'm going at with this is the end of the story for me. So for my friends is that they like a bunch of idiots got drunkenly kicked out of a movie aimed at like 14-year-old girls. But the end of the story for me is I was now sitting alone in a movie theater eating popcorn, watching a movie surrounded by a sea of 14-year-old girls. And I felt very awkward. Right? Right? It's weird. It's weird. Anyway, it's a true story. It's important to read the whole email, is what I'm saying. When you get an email, you want to read the whole email, not just the top part. Because I would have not gone. You know, that's not my thing. I don't want to get drunk and trash a movie. But so I probably would have declined to join. But I was like, I haven't seen Jay and Alex in a while. That'll be fun. It was not fun. Not for me. I think they had fun. I didn't have fun.
30-year-olds to go like, yeah, we're going to get wasted and harass people at a movie theater.
You know, I couldn't tell you. Jay is an idiot. Now he's a cop, interestingly. But he wasn't at the time. He wasn't at the time, actually. But yeah, he's a member of our Royal Canadian Mounted Police. And I must say, like a very good one. He's a really good guy and a really conscientious guy. But I think his getting drunk and watching Twilight days are probably behind him. But probably also should have been behind him before. Does anyone know the year Twilight came out? Let's look this up now. I want to know how embarrassed I should be. The answer is probably rather embarrassed. That's not how you spell that. 2008. Okay. In my defense, that's a little bit better. How old am I in 2008? I'm only 20. Okay, in 2003 I'd have been 20. Okay, so they were only 25. So that's a little better, right? It's a little better? Anyway. Yeah. What year were you guys born? Oh my God.
So it's 1 year older than you.
Oh man. I'm old. Okay, let's carry on. I'm ready to undergo mitosis and then just die. So anyway, where was I? So glad I record these lessons. This is going to be a good one. Another one for the record books. So anyway, in mitosis, the cell divides in two. Yielding two what we call daughter cells. Now, why not son cells? I don't know. Couldn't tell you. This is what we call them. We call them daughter cells. So we call the original cell the parent cell. And interestingly, the parent cell isn't gone. The daughter cells are half that. This is not how humans reproduce. Right? Your parents didn't make you by splitting themselves in half, and now there's two of you. Right? That's not what they do. But this is exactly what cells do. one copy of the DNA, because remember we made a copy, ends up in each daughter cell. That looked horrible. Now, if you're like me, you can see a potential piece of confusion or a potential problem with this kind of simplified version we've drawn here. Right? So we said at the end or near the end of interphase, we go through what's called the S phase of interphase for synthesis. We make a copy of the DNA. Then we said after interphase, the cell actually enters active reproduction, a process called mitosis. And we said that in mitosis, we split the cell in half, and we end up with two cells, and one set of DNA ends up in one, the other set of DNA ends up in the other. How's that feel? Pretty common sense? That's actually the whole process. Yes, sir? Yeah, of course. The only thing we want to do today is say, well, how do you get from here to there? What's that actually look like? And in a couple of days, Will and I use a microscope, and a couple of days after that, we're going to look at some preserved specimens and actually try to find the cells in those preserved specimens that are actually in the process of reproducing, which can be pretty frustrating because they're not like nicely drawn like a diagram, but is also really cool when you get it effectively. What might make this challenging? I mean, what must also split in half before the whole cell splits in half? The nucleus, very good, with the DNA, right? And it has to split apart in such a way that we end up with one copy exactly going into this cell and one copy exactly going into that cell, which you can imagine be kind of tricky, right? Anyone ever moved houses before? I moved houses a few times in my life. What do you do before you move, before you pack up? Or before you move, what do? You pack up. You take all your stuff and you put it in boxes. Why? Sorry? Yeah, way easier to transfer, right? It would be really hard to load everything on the truck if you're just like, there's a stuffed animal, there's a shirt, there's what else do people have there's a plate. Oh, look, it's a cup, right? Like you'd be at it all week versus here's a box of cup, here's a box of stuffed animals, here's a box of board games, right? That's how you do it. So the cell does something similar. So we're going to do this again. We're going to do mitosis this time in detail. Toby, can you go find more paper?
Can I also get another one too?
God love you, Toby. Thank you. Okay. Just ask for lots. Yeah. Mitosis happens in stages. All right. So the first is what we call prophase. Oh my.
Sorry.
No, that's okay. That's okay. That was lovely. I enjoyed it.
There's also anaphase.
Oh, good. Prophase, metaphase, anaphase, and you got the last one, telophase?
No, I didn't hear that one.
That's okay. Okay. In prophase, what's going to happen is this. We'll draw it. So it starts out, like we said, with the DNA. Oh, geez Louise. With it kind of looking like that. And by the end of prophase, what it's going to look like is this. The nucleus is going to have dissolved. And the DNA is going to end up looking like the way you've probably seen DNA shown in diagrams before. If you've seen DNA shown in diagrams before, which is these little kind of X's like that. And we're going to see the forming of a couple of structures here. Okay, so during prophase, the DNA coils and supercoils into tightly packed chromosomes. That's what you see here. Have you ever seen, I don't even know if you have, does anyone have like a grandparent who still has like a phone hanging on the wall in their kitchen? And it's got the cable. You seen a telephone cable before? How it coils like this? Why does it coil like that?
Oh, is that like the RNA?
Yeah, in this case we're talking DNA, but yeah. Why does it coil like that? Because you can stretch it out. If you take a telephone cord this long and coil it, it'll only be like that long. Does that make sense? In fact, we'll show that. We'll show here we have some DNA, and we'll have it be fairly long. There's a piece of DNA, and when you coil it, it's going to get really short. It'll only take up like that much space. Does that make sense? And then what we're going to do is actually coil that again so that it's like making coils like that. And the result is that it takes up way less space, but also that it's protected, which makes sense because the next thing that's going to happen is that we're going to get rid of the nucleus. So this is our DNA. This is coiled once, and then this is supercoiled. And in fact, it actually gets coiled like 3 more times. If you take grade 12 bio, you look at all the steps of this. It's wild. It basically gets packed up in a big box. It basically just gets tightly coiled so that it can be safely transported. For this course, we don't need to get into that detail. But this is to keep it separate. You can imagine that this is like having loose pasta or something, right? Or like, you ever have like headphones with cables, and no matter what you do, every time you like put them away, they end up in a big tangle? Yeah. Whereas, if you wrap them nice and neatly and Velcro them, then they don't, right? This is the equivalent of wrapping them up really nice and neatly, Velcroing them, and then maybe getting 10 pairs like that and wrapping them all up together and Velcroing, and then getting 10 more and then putting them all in a box. Does that make sense? It's packed up. Not very useful, so the cell can't use the DNA when it's like this, but that's okay because it's in the process of packing it up and splitting it. Okay. Then next, the nucleus dissolves. Why does the nucleus have to dissolve if we're going to do this? Well, because we've got to get some of the DNA over to one side of the cell and the rest of the DNA over to the other side of the cell. Otherwise, when it splits in half, all the DNA would be in one cell and the other daughter cell would have no DNA and wouldn't be able to function. So the nucleus dissolves, and these are called centrioles. Centrioles form at each end of the cell. And we'll learn what the cell centrioles do in the next stage. So that's prophase. Prophase is all about packing up the DNA and then getting rid of the nucleus so it's ready to be moved. And so to make you do all this drawing, I used to do this with a handout where I gave you the drawings, and I found that it really does help to draw it yourself. It's annoying. I totally grant you that it's annoying, but I really do believe that it helps you kind of understand it a little better. It does sound that way, doesn't it? Who are they?
Your test writers.
Okay, so tell them that if they have time to be messing with the test tape, then they're obviously done with the test. And then get back to class. Do you want me to say that? Are you comfortable saying that? Toby's comfortable saying that. Toby doesn't get it.
If they don't, I can just pop out.
Yeah, or hit them. You pick a medium-sized one, make an example of them, then they'll fear you. Again, I'm so glad I'm recording this. I'm obviously kidding, and I would never, ever actually suggest that. Obviously, I think it's important to be respectful of everybody's, personal space. Okay. Just so we're clear there. Very important. Okay. After prophase comes a stage called metaphase. Oh, I like that. It's got a song. I think every phase should get a song. That was kind of soulful. Like maybe the next phase could be like death metal. I don't know. You can make it happen. I have faith in you. Yeah.
Wasn't mitosis stage 2?
We're talking about the stages of mitosis now. So now we're talking about mitosis itself, and mitosis happens in stages. Good question, but you're right. Overall, mitosis happens after interphase, and now we're breaking into mitosis and saying, well, what happens there step by step? Great question. All right, so in metaphase, we're going to mention something that started to grow in the previous one. So here, in the previous stage, the centrioles begin to produce something called spindle fibers. What's a spindle? Does anyone sew? Or does anyone fish? They call it a spool, but it's the same idea. Spool, a spindle, same idea. It's like a thing like this that can cast out a string or a rope or a fiber and then bring it back. So if you wanted to call these spool fibers, I mean, I'd mark you wrong in the test, but it means the same basic thing. Would I mark you wrong? Yeah, I would. It's not what they're called. So in metaphase, what we're going to get is this. The chromosomes of DNA are going to line up at the center of the cell, like this. You know what? I'm going to have them alternate in colors and try and stop me. So again, these are those tightly packed, coiled and supercoiled DNA chains, like that. And by this stage, the nucleus is completely gone. It's not present. And the spindle fibers are actually going to reach out, and they're going to attach to the chromosomes, just like this. And when I say just like this, I mean, the truth is the spindle fibers actually shoot out everywhere. They don't know in advance where the chromosomes are. So they're actually shooting everywhere through the middle. But the ones that come into contact with the chromosomes attach to them. There's actually all kinds of other ones that are just attached to nothing. But if I drew too much of that, it would get messy. And they attach to the chromosomes at this little piece inside each chromosome called, and wait for it because all these words sound exactly the same, it's super annoying, at what we call the centromere. So if we zoom in here, we can see there's our DNA. Why'd I draw that so big when I'm not using it all? I don't know. And it has this central structure called the centromere, and that is where the spindle fibers attach. So the spindle fibers connect the centriole at each pole to the centromere of each chromosome. I really hate those words. I wish, usually in science, I think they actually come up with pretty good names for things. This, I think, is an example of a swing and a miss. I think they're confusing, and I don't think it's easy to follow. But, hey, that's okay. What do you think they're going to do? Again, we said a spindle or a spool is like the thing on a sewing machine or like on that. So they've shot out and they've connected to these chromosomes. What's going to come next? Why to do that? Well, you said we have to get half the DNA to one side and half the DNA to the other. Yeah. Yeah. First they spool out the spindle fibers, then they're going to spool them back, and they're literally going to be little fishing lines. It's kind of cool, right? So, in this stage, the spindle fibers connect to the DNA, which, remember, when it looks like this, we're calling chromosomes. I totally colored this like a biology lesson, not like a mitosis lesson. That's disappointing. I colored them as if it's maternal and paternal. That's okay. Don't worry about it. We'll work with it. Damn it. That's okay. We'll correct it later. See, I was in bio mode. I've taught grade 11 bio like twice in a row since I've taught this. That's okay.
Now they're just more colorful.
That's right. Needlessly so, but that's okay. At the centromere. So the centriole connects to the centromere of the chromosome via the spindle fibers. Okay. The DNA is lined up at the middle of the cell. And again, these are the chromosomes. You can write just one of those two words if you want. Oh, that's so irritating. When I teach you genetics, this color code is going to make a lot of sense when you take grade 11 biology. You're going to be like, oh, that's why he colored them two different colors in grade 11. But I should have done something different. But that's okay. We'll fix it. Whatever. We'll just explain it. Makes the diagram a little less good, but if I'd printed it, it would have been black and white anyway, right? Okay. After that, like you just said, the whole point of this is we've shot out a fishing line. We've caught it. We've hooked what we want. And once you throw out a fishing line and you hook what you want, what do you do with it? You reel it on in. So what's going to happen in the next stage? Now imagine this gets a little gruesome. Let's say hypothetically we were fishing for, I don't know, Heimple. And let's say that Toby's over there and hooks on. I'm over here and I hook on. We both start reeling in real good. What's going to happen? Yeah. I mean, obviously in reality, Isabel would just like break the line or something or like come and fight us. But you can imagine that in some hypothetical world, ripped in half, right? That's what's going to happen here. We made a copy of the DNA, and as such, half of it's going to move to one side of the cell, and the other half's going to move to the other side. So, oh, I didn't write the title. This is called anaphase. And in anaphase, the spindle fibers that have come out from the centrioles start reeling in, and they actually split the chromosomes in half. So the original chromosomes were stuck to a copy of themselves. And now, oh, I only drew 4, didn't I? Yeah. Does anyone know how many chromosomes humans actually have? I've only drawn 4 because it would be completely miserable otherwise. But does anyone know? You actually have 20. You actually have 46. 23 pairs of chromosomes. Humans have paired chromosomes. We'll talk about why if you take grade 11 biology. So you actually have 46 chromosomes in every one of your cells. That's a lot of DNA. Your DNA from one cell, from just one cell, and you have trillions of cells. If you uncoiled your DNA completely and stretched it out, just one of your cells would have about 1.5 meters of DNA. And you have trillions of cells, which means there's enough DNA in you to go like to the sun and back a handful of times, which is crazy if you were to stretch it out. Isn't that weird to think about? I mean, it's not just weird to think about. I think it's actually like impossible to think about. So the chromosomes split. With one copy of each chromosome going to each pole. And the reason that's able to happen is what is pulling on them? The spindle fibers. Which you'll sometimes see called astral rays in some sources. I kind of like that name even better. It's like space rays. It's a good name, right? I think it's a good name. So just to show what would have happened here is the spindle fibers actually connect to centriole like this. and the split happens at the centriole, and one copy of the DNA, because this is from when it was copied, even though I screwed up the color coding, gets pulled one way while the other gets the other. So this here would be one copy of the chromosome. And this would be the second copy. And so when it splits, one copy goes to one side and the second copy goes to the other side. You can almost think of the chromosome as being stapled to a copy of itself at the centriole, or at the centromere rather. All right, and now what can the cell do? Now it's got a perfect copy of its DNA on the left side and a perfect copy of its DNA on the right side. We are one section away from being done, team. Are you excited?
I don't even know how. I'm going to be so serious.
That's okay. We're going to talk our way through this like 4 or 5 times. It's confusing. There's a lot going on. And then you're going to build me a model of it. I think that helps. Oh, we do it. That was a really interesting one. She chose to do it as a fashion project, where it was a series of dresses with the steps of mitosis painted on it. Interesting. I thought it was creative. I liked it. And it got all the science right, so that was good. I had people do all kinds of things. I had somebody make an animation for me once. I had one group do like claymation for me once. That was really fun. I had one person once make me a podcast about it, which is not what I'd expected, but it was really good. They got all the marks too. Okay. So then in the last step, we get what's called telophase. And this is where it's actually going to split in half and you're actually going to end up with 2 new cells. So in telophase, we start to get the cell actively dividing. That didn't look very good. Let me try again. We get the cell actively kind of squeezing in the middle, like that. And we get the formation of a new nucleus around the chromosomes. So here we have green, green, purple, purple, because I screwed it up. So annoying. Okay, so a new nucleus forms on each side of the cell. And the cell starts to pinch in to divide itself in 2. This is, have you ever seen like a soap bubble where if you squeeze it in without breaking it, you can divide it into 2 bubbles? Ever seen this before? Believe it or not, soap bubble is actually the exact same material as cell membrane. We think of soap bubbles as being really delicate because they're really big. Cells are really small, so that same material there is actually very strong. But it's the same idea. It simply squeezes in and will divide in 2. This is called the cleavage furrow. You don't know what it means to cleave something? Like a meat cleaver is used to cut meat in 2. I spent years perfecting my diagram of telophase so it doesn't look like breasts, because if it looks like breasts and then you write cleavage furrow there, then you really lose the room, let me tell you. Learn that from hard experience. Yeah. Yeah. Anyway, it's frustrating to me that the term is cleavage furrow, right? Like I wish it had a different name, but it doesn't. That's the name. So there we go. I didn't invent it. Well, again, to cleave means to cut something in half. And a furrow means to, like, make a shape like that. Does anyone ever, like, you furrow your brow, right? So, like, you squeeze the brows together. So, this is, by squeezing the membrane together, furrowing it, you're going to cut it in half. Cleavage. But again, we think of the word cleavage meaning something different. So, that's right. Anyway, moving on. So, a new, oh, for God's sakes, I didn't write the key word. A new nucleus forms. Yeah, sorry about that. A new nucleus forms on each side. I'm just going to rewrite that. I'm sorry. I'm sorry. Don't judge me. Okay, and then the cell furrows, i.e. pinches in, in the middle, preparing to split in two. And then that's the end of mitosis. This is a stupid little technicality, but mitosis is the process of one cell. So mitosis ends right as it's about to become two cells. And so finally, we define cytokinesis as two cells separating and moving away from each other, which again makes sense because cyto means cell and kinesis means moving. So whatever, it's cell moving. That's kind of dumb too, right? I didn't make these names. Technically, this stage isn't the last stage of mitosis. It's what happens after mitosis. In practice, it's basically the last stage of mitosis. Bless you. The two daughter cells separate from one another. Now, in all likelihood, that will have gone a little bit in one ear and out the other. I would expect. All right? That's usually how it goes, especially on Monday, after a weekend. A lot of words, a lot of terms. We're going to get it in our notes now. We're going to talk through this plenty of times. The big takeaway, if all you take away from today is you have this written down and then you understand the basic thing, which is you have a cell, in the middle of the cell is the nucleus with the DNA, and to reproduce, what cells do is first they make a copy of the DNA, then they move one copy to each side of the cell, then they cut themselves in half. If you have that, you have all the important details. Does that make sense? The rest of this, the specific steps, sure, I care that you know that, but not really. You know what I mean? I'm not so much a memorization guy. And once you really have that down, the steps actually make sense, right? If the cell's going to split in half like this, what's going to happen? Well, the first thing that's going to happen is we're going to pack up and get ready to move, right? And then, just like you'd have to hire a moving truck, the cell has to have something to move stuff around. What it has to move around are these things called spindle fibers, which are literally have made something called microtubule. They literally are like little fishing lines that shoot out from each end of the cell. They grapple onto everything, and they pull it to each side. At which point, we basically build a new nucleus. We build a new house for each of the two cells' DNA, and then we cut in half. Does that make sense? And we'll draw this diagram. So if at the end of mitosis, we had the cleavage furrow forming like that, that looks like absolute trash. Let's try that again. Okay, and what's going to happen then is we end up with two cells, and these two cells can then just go live a happy cell life. What do we call a cell living its happy cell life, by the way? What stage is it in? That's okay. You're allowed to forget. Check the top of your note. So two daughter cells, which can enter regular cell life, which is interphase. Good. So fundamentally, thinking of like how animals reproduce is a very bad example of this, right? Yeah. How's that? Is that better? Good. Sorry about that. So yeah, you can't think of cellular reproduction using kind of human reproduction as a model, right? This would be the equivalent of if we got really big and then just cut ourselves in half. Is it true that some worms, you can just cut them in half and both halves grow into a worm? Is that true, or is that like an urban myth? I don't know. But that would be a good example. It sounds like an urban myth. Sounds like something that was said on my playground full of stupid kids who had no idea what they were talking about. I have no idea if that's true or not. I should look it up. I never really studied annelids. I don't know much about worms.
I don't think it happens because I saw
Anyway, what I know definitely doesn't happen with people. So don't you go cutting yourself in half. All right? Doesn't work that way. Because even some animals can regrow limbs, right? Like, some reptiles can like regrow a tail or an arm or something, right? But again, you know what can't do that? People. But again, big takeaway for today, when a cell wants to reproduce, it gets bigger, it gets bigger, it gets bigger, and it cuts itself in half.
Mac and cheese with a dinner roll for $6, tuna or egg salad sandwiches for $4, and fries and small beans $2.50.